Does this work? Yes. So you have to be very close. So hello everybody, thank you for joining this session on food production for exploration. So we have two very interesting speakers today, which will be followed by a panel discussion. And the aim of the session really is to help us, ESA, understand what we should be focusing on in terms of food production for exploration. Where are the gaps in our program and how can we address them better? So I will invite our speakers, and then at the end we will have a Q&A session and a panel session. So please keep your questions for the end. And our first speaker is Åsa Berggren from the Swedish University of Agricultural Sciences. Thank you very much.
Thank you very much. Let's see, I guess this is our thingy to use. Yeah, is it working? Yeah, I'll find out soon. Hi, everyone. Nice to be here and nice to see that many are interested in food. So am I, but I'm actually a biologist and ecologist. So I'm going to talk a lot from that perspective here today. The work I'm going to talk about, we are several people that we've been working with. It's Annette Brun Jensen, David Copplestone, Roberto Guidetti, Martina Hea, and Paula Piccia. So, we are planning to go quite far, aren't we, to different planets and even long distances. So, obviously, we need something good and nutritious to eat on that way. So we will need to bring food. But the thing is, we need so much food, so it's very hard for us to bring all the food that we need. I think there are estimations that you need about approximately 1 kilo of dried food per day per astronaut. And if we're thinking about 4 people going away for 3 years, we need about 4 tons of food for those people. This is not an amount we can really bring with us. It's too much, too heavy, and it's very, very costly. So we have to think about another way of getting food to all these people that are going, because maybe not only 4, maybe there are lots and lots of people that are going to go now in the future. So this is a bit of a slightly strange. I'm asking ChatGPT to do me pictures of insects and plants, and this is what they come up with. Usually they just give me lots of fish. I don't know if they, and I ask for insects, I get basketball-sized insects. But anyway, so we are going to see something new in the future. And when we are going really, really far away, of course, we have to utilize the resources where we're going in a way. We can't bring anything with us for the stuff that we will produce to eat. So we have to bring some stuff with us, some animals and some plants or seeds or youngsters or whatever. But it's really, really good if we can utilize the resources wherever we go. And how are we going to manage to do that? We have to think about new ways of doing this, because obviously this is stuff we have never, ever done before. And don't forget, the food is super crucial, because we, as people, as humans, we can't live without food. And now I'm getting into the insect side of things. So these are some examples of insects in human food today. So maybe you were expecting just some photos of some whole insects, what people are consuming, and people do. In some countries, it's a tradition to eat whole insects. But if we look at more in Western countries, this is not the way how most people eat insects, because insects are a part of different food. They look like food you get to shop and to eat in the store. So here we can see we have biscuits, and the stuff in the upper middle there is actually like mince that you eat with pasta and stuff like that. We have burgers, we have chocolate, we have chips, bread, and whatever. So there are muesli. I've seen lots of chocolate lollies. I've seen smoothies. There are all kinds of stuff that you can buy and eat in many European countries today. And they all look and taste like stuff you eat for normal dinners and snacks and so on. So we have it already around us. And if you look at the rest of the world, it looks similar or even more common to eat insects around the world. And actually, in many parts of the world, this is common that you have in your diet. It's not the main part of your diet, but you could be eating it several times a week or part of other meals. So there are estimations about 2 billion people eat insects today. And for them, it's not weird. It's normal. And they eat it because they think it tastes good. And there are also estimations saying that this insects production and eating insects will increase all over the world. And there are huge investments around the world now building rearing facilities to increase the production of insects for food around the world. So there are lots of money being invested in this area at the moment. Oops. So currently, most of the facilities, if you look at the numbers around the world, actually really small facilities where you rear insects. There are more like family companies and so on. But now these really large facilities are starting. And they are enormous, some of these. They are sort of flight hangar sort of style or sizes, where lots and lots of insects are produced in a sort of more automatic way now. So why do we think this is interesting at all? Well, there are a fair amount of studies being done, and the studies show that for the different species, insect species they've been looking at, they have lots of interesting nutrients for humans. Many have a very high protein content with the suitable amino acids for humans. Many have a quite high fat content as well. And some of the species have fats that are similar to fish that we know are healthy for us. They have vitamins and minerals and fibers that are also interesting for human nutrition. But there are other aspects also of insects that are sort of putting them a bit above some of the animals that we usually eat. So in this figure, we have compared different animals that we eat. We have crickets, and we had chickens and pork and beef. And the staples that you see there is how much feed you have to put into these animals for them to grow. And when they have grown, you can eat part of these animals. So if we look at cows, for example, we have to push in around 25 kilos of feed resources into cows, and we eat about 1 kilo of those animals. The rest is discarded, and actually it's a bit of a problem in some cases, because that's something you have to deal with, and it's a cost or an environmental cost. The other thing that is interesting with insects is that because of how they function and how they have developed, they can actually eat things that we can't. So we as humans, we don't compete with insects for the food that we can eat. So insects can eat what we regard as waste. So that is an additional component which makes it interesting when we think about system where we have waste that we want to get to use for something good. So all this together makes it interesting for us to look at how is there a potential for insects to use as food and maybe also waste converters when we think about doing these really, really long trips in the future? So, but obviously, this is a very, very complex question. So we have worked with this and sort of going at it from many different disciplines because there are so many questions. So it's not just one discipline that can answer these questions. We have to be researchers from many different fields. So we have different biologists, ecologists, human physiologists, food technicians, and so on. And we've been working in 2 different groups. So we've been supported by ESA on a topical team that we've been working with, but we've also been supported by ISGP in a scientific subcommittee and working with the questions there. And the way we've been looking at it is we have this biology side, so we have to know how would different insect species actually work in an environment that is totally different from the environment here on Earth. You can see it as starting to look at something that is totally new. It's like for the first time asking, how would humans work or function in space? So we're almost there now with the insects. We're starting to ask these questions. How would they work? How would they survive, develop, reproduce in a space environment? At the same time, we have to understand more about the nutritional value of different species. So we are talking about insects here. But don't forget that insects, there are so many insects around the world. It's like thinking that all insects would be the same. It's like a horse and a mouse. Of course it's not the same. There are so many, and they're also evolved in different environments and eat and function in different ways. And we're also interested in, if we make food from insects that have been in space, how would that work? How would the food taste? Would it be different? How can you store that type of food? And so on. So what we did was to do a big review where we have examined what do we know about different insects? How would they fare? How would their food be? How would the nutrition be? To just get a baseline on where is the science today. So we have done a fair amount of work on this, and most of the work that I have been involved with myself has different ecologies in focus. So we have been looking also at separate studies looking at how the insects interact with their environment, where they are. We've been looking at circular systems, if insects are going to be a part of a systems where we imagine that you have plants and maybe the insects can eat plant parts that we can't eat, and they create their poo is called frass, and that could be used as a fertilizer and so on. So maybe they could be a really nice part of a system that we produce not only food for humans, but other resources as well. We have been investigating different pathogens and diseases because insects have them as well, and looking at different behavioral issues because we want animals that are happy and healthy and live well and not aggressive or being mean to each other or whatever. They behave exactly like all other animal species on Earth. And some of the studies we've been leaning against so far are a fair amount of studies that have been looking at how do actually insects survive on these different byproducts. And on this, in this study, the project was, or the study was focused on looking at different, what is sort of regarded as waste. So that could be food byproducts, it could be when you do beer, you get all this leftover stuff you just toss away. Or if you grow wheat you maybe not use anything left of the plants. So there are lots of things just being wasted around the world today that is interesting to try out as feed. And you see the orange arrow up there, and that is pointing at chicken feed. So chicken feed, what we give to chickens, is actually super nutritious feed. Chicken feed we can eat as humans, and we are giving our chickens stuff with such a high quality, so it's actually a waste if you think about how we use the resources on our planet. And here we see that some of the stuff that was tested here didn't work well. The insects lived and grew not that well, but there are obviously some different weeds or byproducts that could be interesting to have in these systems. So some of the general findings we have are, first of all, and not surprising, there is a massive knowledge gap on insects and insects in sort of space environments today. There is not much known, which is sort of interesting in one way, because then it's sort of an open field if you want to do research on this. But it also means that there is a massive amount of work to be carried out if this is going to be reality. We see when it comes to insects, there are very few long-term experiments in space situations. There are some, but they're very short. There are hardly any experiments carried out, so it's just looking how things are going or living, so it's not really any experiments. There are very few species that have been tried. We talked about previously today, in many studies now are looking at the difference of men and women responses to different factors. So it's not strange that no one has really looked at that when it comes to insects, but obviously that's the same thing there. Or different life stages. I mean, how do young insects fare compared to old insects? Because we have to have all sexes, all life stages, if we're going to produce insects. And not strange either, there are no studies that are complex at all. It's very, very simple studies being carried out. And we know that factors can interact. Gravity and radiation together can have totally different impacts on organisms than just one factor itself. So nothing like that is done either. When we look at food production, the stuff that's been done so far is mainly on plants, which is good, of course, but there are very few animal studies in space. When it comes to insects, there has been done experiments on insects, but then it's mostly been on fruit flies, and fruit flies is this sort of standard model for looking at biology in many different respects, so it's not strange either. But we don't eat fruit flies, or I guess we could eat fruit flies, but you have to have a massive amount of fruit flies if you're going to eat them. There are some effects of gravitation and radiation that has been seen, but these are just a few sort of, if you're looking at sort of trying to get the picture is very, very hard because they're sort of scattered around. One has seen slower development for some species, but it's not a general thing. Increase in number and size of eggs for some species. Fewer and hatched and developed to maturity for some. There have been some injuries and anomalies reported in the morphology, in the development, especially from radiation. Some effects on the lifespan of how long the insects live. And also some have reported a reduced function of the immune system and muscle loss. So nothing of this is very spectacular, and this has been seen also, of course, in humans and other species as well. But it's very, very hard to get the really good general picture here. What we can say is that insect species do respond differently. Some species doesn't seem to be that affected by some of these factors, like radiation or different gravities. And some species seem fairly robust against these major changes from where they have been developed and evolved here on Earth from the other types of conditions they would fare if they go up in space. So from our findings so far, we conclude that we think that some insect species might be able to develop and reproduce, because we don't want them just to survive. They need to reproduce so we get big populations that we can harvest and eat in space environments. And insects have the potential to be part of a bioregenerative life support system. So from the knowledge we have so far, we think that there is a potential for insects to offer nutritious food for future space travelers and colonizers. So what we are doing at the moment, we are doing experiment here on Earth in different locations. And we are focusing on the species that are approved for consumption in Europe today. There are 5 different species that are approved and currently reared and produced in Europe. So we're looking at different biological processes there. So we're looking at the effects of microgravity and radiation on how well eggs hatch of different species, the mortality and the egg morphology and so on. We look at how these different species are developing. And we're also looking at how well they reproduce. And this links on to the food part, because we need to know what happens with these insects. Even if they survive and we get lots of insects, how does that affect the nutrients of the food that we're going to eat then? So does that change or not? Or not that much affected? Or if it changes, how does it change? And how does it change for different aspects that are really important if you're going to eat them? Does the flavor becomes different? Are they still tasty? Or how can we store them? Does the storage change if they are radiated and so on? There are lots of these aspects are really important. And all these studies are linking to each other. So we utilize the labs that we have, the species that we are rearing, and the competence of the teams. So that was what I had to say. So thanks very much for hearing me out on that. And I'm super interested in working this way with different types of competences, because I know what I can do. I don't want anyone else like me. It's super boring. So I want to work with someone else that knows something that I don't know. So anything you can think of or have an idea or so on, so just come up to me and talk about that. And all the work we're having is just free to download from ResearchGate. And thanks to ESA for supporting our topical team. Okay, thank you.
Thank you. So we will do questions after the next speaker. Thank you very much. And our next speaker is João Garcia from the University of Wageningen, who will talk to us about cellular agriculture.
Yeah. Thank you, Christian. First of all, thanks to the organizing committee for giving me the chance to talk about cellular agriculture, or cultivated meat, which is going to be the strong point of this talk. So a bit of background. I'm working at Wageningen University, which is a university in the east of the Netherlands, and one of the best universities in the world for food science and agricultural research. I'm in the bioprocess engineering team, which pretty much uses any cell type that you can imagine, microalgae, yeast, bacteria, to produce any type of target component that might be interesting, from biofuels to therapeutic molecules to food. Within the group, there's a cellular agriculture team, which is where I work, and we're also part of a wider Dutch consortium called Cellular Agriculture Netherlands, which was funded by the Dutch government with 60 million back in 2022. And it's a consortium of universities and companies that aims to promote cellular agriculture research and education in the Netherlands. Oh, I think this is the wrong presentation. Sorry. Indeed, first I wanted to start with asking you, can you guess what this picture is? So I give you two options, since we're at the space conference. It's either an image of the James Webb Telescope or animal cells or muscle cells growing on a plant. I'll let you know at the end of the lecture. So yeah, we bypassed this. So why do we work on cellular agriculture? Our food system is not sustainable, as we've heard before. So about 50% of our habitable land is used for agriculture. 77% of that is used for livestock production. And from all that land, we only supply 18% of the global calorie supply. So a pretty inefficient system. Our agriculture also contributes to 22% of carbon emissions or carbon footprint. It's also a major cause of biodiversity loss. So here on the left, we see regions with a higher biodiversity loss in yellow, which is somehow correlated with areas where there's a higher livestock density. And this number is pretty scary. So of all the mammals on Earth, 94% are livestock and only 6% are wildlife. And also pretty striking numbers. So on green, you have the natural lifespan of the different animals. Red is the age they're usually slaughtered for human consumption. And these numbers in red are the number of animals that are killed per day for human consumption alone. So it's obvious that we need alternatives. And we, of course, have plant-based, which is quite widespread already. But two new technologies came up recently, precision fermentation and cultivated meat. And they're part of what we call cellular agriculture, which produces conventional agricultural products using cell cultures by leveraging synthetic biology or tissue engineering approaches. So in precision fermentation, what we do or people do is that we modify microorganisms to produce molecules of interest. That can be milk proteins, flavor compounds, etc. And just an ad from some colleagues of mine, two assistant professors in our group that are working on precision fermentation. So they have projects running on uncoupling production of target compounds from the growth of the biomass of the bacteria. They also aim to enhance protein secretion or improve protein recovery. But cultivated meat. So I'm a biomedical engineering by training, so I worked in the medical field before, and I was using cells from patients. I would grow them outside the body to then produce, well, the aim was to produce organs or tissue that could then be implanted back into the patient's body. Of course, this doesn't really happen at the moment. What we do now is that we grow cells outside the body and we use them as models to test drugs. Cultivated meat is pretty much the same. We take cells from an animal, we grow them in the bioreactor, and we produce food products, steaks, burgers, meatballs, whatever you want. It's not sci-fi, so there are already some cultivated meat products for sale. So Hong Kong, just last week, a restaurant launched a product. It's a sort of a cultivated quail, which is similar to chicken. This is same product or similar product being sold in Singapore, And also in Singapore, you can buy cultivated chicken on a butcher shop. This is pretty cool, but it's not widespread yet. It's still pretty expensive to produce these products, and we cannot really produce them in scale. There are several challenges or technical challenges because, of course, societal challenges are much more. But the technical challenges are the media. So the media we give to these cells is still pretty expensive. Our cells are not optimal because they're primary, so that they're not standard. They might stop growing after a while. And of course, we don't know how to grow these cells in large bioreactors. So we're talking about going from lab scale to, I don't know, fermenter, beer fermenter type of scale. And this is what we're doing in our group. So if you look at the production chain, we're very much interested in media development. So we really want to bring the costs down. And one of the reasons is that media accounts for 90 to 99% of the production costs of cultivated meat at the moment. And this is the composition of media. So we have a carbon source, which is usually glucose, nitrogen source, which is general amino acids for protein production, of course. Then we have some salts, vitamins, and growth factors and other molecules that are usually the most expensive part of the media. So we just hired a PhD student which is going to explore different components that can be used for media formulation. So he's going to be looking at hydrolysates from different plants, bacteria, microalgae to produce these media components. We're also going to look at agricultural and industrial side streams. But more importantly, we're looking at transitioning from pharma-grade ingredients to food-grade ingredients. As I said, cultivated meat was transitioned from the tissue engineering field, which is a medical field, where the profit margins are quite big. So if your media is expensive, doesn't matter. Your profit is even higher. But in food, we cannot afford that. And an example of that is this picture here. So this is, say, a component supplier, Merck or Sigma, and this is the price of baking chocolate. So you would pay 1,500 euros for 200 grams if you would buy the chocolate at Sigma. If you do buy the same amount at a Dutch supermarket, you pay 4 euros. So they are pretty much the same component, the same material, but they have an immense difference in costs. So this is sort of the transition that we need to do also in the cultivated meat, and this is proven to work. There's a cultivated meat company in the Netherlands that has managed to decrease the cost of their media by 90% by replacing their components from pharma grade to food grade. We're also very interested in cell line development, and why? Because cells are the building block of cultivated meat. So as I said before, we usually take cells from the animal, we call them primary cells, and these cells will grow for a while, but then they'll sort of stagnate, become senescent, and die. They keep a very good differentiation capacity, and that's why they're good, but we want cells that are actually immortalized. So these have a higher proliferative capacity, so they're able to divide pretty much forever. They have an efficient metabolism, and they also keep the differentiation capacity. On this, we're looking at the octopus as a model organism. So I'm from Portugal. Octopus is very relevant for us in terms of gastronomy. It's also very important in Spain, Italy, Greece, Japan, and South Korea. And it's becoming increasingly important also in countries like the US and China. There's an increase in demand. We also see a stock depletion of natural sources of octopus. And some companies like Pescanova is looking at producing these animals in aquaculture conditions. This is seen as not very ethical because octopus are very intelligent, sentient animals. They do not like to grow in confined conditions. And in fact, in the US, in the state of Florida, they banned, the government banned aquaculture of octopus. Last but not least, because I'm a scientist, I'm very interested in the octopus innate regenerative capacity. So once the octopus is injured in nature, for instance, the arm will grow back fully functional in a matter of weeks. So we also just hired a PhD student 2 weeks ago, Joanna. She's going to be looking, in collaboration with a group in Naples, she's going to be looking at which cells are responsible for this regeneration mechanism in the octopus. We're going to study these cells, see where they are, and hopefully isolate them in vitro and produce octopus muscle in vitro. But what about space? I think that's why I'm here. So if we go back to what are the nutrient requirements for space, so is a document that you can access, it's produced by NASA, talks about the nutrient requirements for deep space exploration missions. These are how you calculate the energy requirements. So we need about 45 to 60% of carbohydrates of all this calorie intake. 20 to 35% needs to be fat, and protein, 1.2 to 1.8 grams of protein per kilogram of body weight. But importantly, 60% of that protein needs to come from animal, and this is the important bit. So this is, well, how this food system looks currently. A lot of thermostabilized food, freeze-dried food, and a very small amount of natural or fresh food. This works quite well on the ISS because we can resupply quite frequently. But as we think about going to Mars and potentially even further, this doesn't really work anymore because the shelf life of these products, of the thermostabilized products, is 2 years. So it's not that the product will become bad or get moldy or so, but we don't know what happens to the nutrients, or the nutrients will actually start degrading. We also have a reduced possibility to resupply the crew or the ship. If we have to resupply everything in one go, then we also have a very high launch and volume mass. And we know that packaging at the moment accounts for 50% of the total food weight. So we need to think differently, and this is sort of the characteristics that we would be looking at for a new food system in space. It needs to be varied, so of course, plants, insects, meat. It needs to be safe, reliable, easy to use. We need to minimize resources, and need to be tasty in the end. So we've been looking at plants, microalgae, and now recently into cellular agriculture and cultivated meat, specifically because of that number, because animal protein is still required for deep space exploration. If you look at the timeline, so cultivated meat is not a new topic in theory. So the first mention to cultivated meat was actually Winston Churchill back in 1931 that said the current system that we have to produce food is quite inefficient, and at some point we'll be able to grow chicken legs or chicken wings without the animal. In 1991, there was the first patent by a Dutch inventor. 2013, the first burger was produced. And lately, we've been having some FDA approvals and approvals in Singapore. But already back in 2001, NASA was working on a project to grow goldfish and turkey cells to feed astronauts in space. They dropped the project because they thought it would be too expensive. JAXA in 2018, they funded a project that looked at growing meat cells together with microalgae. And the idea was to have a circular system where the meat cells would be fed by the microalgae, then the microalgae would convert the waste from the animal cells into novel foods as well. 2019, there was a project on board the ISS, 3D printing or a bioprinting experiment. And in 2022, ESA launched, say, 3 feasibility projects, 2 with industry, and 1 was a research fellow, which was me. So I was at ESA until March this year, working on feasibility of cultivated meat for space exploration. Now in Wageningen, we actually have, we got a group of very motivated students. They were very enthusiastic about cultivated meat in space. And Annemiek and Melania, they wanted to model a bioreactor for cultivated meat in space. So we gave them these constraints, which are, I think, the current architecture for a mission to Mars, 1,200 days, 4 crew members with an average weight of 82 kilograms. And this is the requirement of animal protein. So here they modeled the cell growth. So in the first phase, we have an exponential increase of the cells, so they proliferate. And then they sort of stagnate because this is what we call differentiation. So it's when the cells, in this case, the muscle cells, they start accumulating protein. So you see the cell number stabilizes, but when we look at protein, there's an increase. So this is how the protein production will look over time. According to these parameters, they found that the bioreactor itself would be the size of about 500 liters, so half a cubic meter. which is more or less a third of an international standard payload rack, so not that big. And they also did a sensitivity analysis where they saw that if they increase, of course, the maximum cell density within the bioreactor, there's a decrease in size, and the same for the growth rate of the cells. 3 other students, Lorenzo, Soleil, and Paul, they were very much interested in using plants as scaffolds to grow meat cells. So what they did here, and this is not a novel topic, it has been done in the medical field, but we use plants, we wash them or decellularize them, so we remove the plant cell out of the plant, and then we put our cells of interest, in this case muscle. So this is, I don't know if you can see, but these are 2 different plants. You can see they have different morphology, and this is without plant cells, and this is after 2 weeks with muscle cells. And you can already see that there are some differences because you see that this plant here, they have a more aligned structure, while this one is slightly more messy. And you sort of see that replicated into the morphology of the cells. So if you look further, this is how the cells look in the first scaffold, very aligned, while in the other one, the organization is a bit more messy. Here you see the shadow. This is the natural vasculature of the plant. This is a spinach leaf, by the way. So yeah, cultivated meat is promising. So if you think about this plant experiment, we're sort of breaking boundaries, so we could eventually create a product that not only provides animal protein, but also the vegetable protein and the carbohydrates that the crew needs. But of course, there's a lot of problems that we need to look at. Ultragravity, so as soon as astronauts go to space, they start losing muscle. So it's pretty counterintuitive that we want to grow muscle to feed them. So we need to see what is the effect of ultragravity on the growth of these cells. Deep space radiation, of course. Regarding the system, yeah, at the moment we don't cook in space, so if you're producing raw meat or raw cell biomass, we need to come up with ways to really cook these products. And of course, we need to think of ways to minimize resource utilization. Just another add up. I also have 2 other colleagues, which is I've been working with Melissa, or I don't know if you know Melissa, but this is circular concept of ESA, of having the crew, some components or compartments processing waste of the crew. So this is microalgae and bacteria, filtering water and feeding new products or new food for the crew. They've been working on downstream processes to improve the product formulation in this system. So happy to connect with them if you would need. Another self-promotion. So we're actually the first university in Europe to launch courses in cellular agriculture. We started this year. We have a course that looks at the societal implication of cellular agriculture. We have a course, which I'm the coordinator of, on cultivated meat and seafood. So we look at the technical aspects of cultivated meat and seafood. And we have another one on precision fermentation. I assume most of you did your master's already, but if you still want to learn about cellular agriculture, we also collaborate with the University of Delft in an advanced course. And in Wageningen, we also have a summer school on space farming. So this is our team, our leader. So the team didn't exist a year ago, and now we're about 10 people and growing. And if you have any interested student, feel free to reach out. And thank you.
Could I ask the speakers to come back? So maybe it's easier if we take a seat. I have a few questions of my own, but I would like to open the floor really to have a discussion with the audience as well. So I will first ask the audience before asking my questions. So I will pass the microphone.
So first of all, thank you. I have to preface this by saying that I'm not a biologist. I'm just a enthusiastic amateur consumer of food. So in one of the slides you showed, you had this vision of like these big glass domes, probably on Mars, growing something. And insects on Earth are important pollinators. If we think about food production like on the ISS, on a spacecraft, this is small scale. Someone could go around with a cotton swab and pollinate stuff, but once we go to sustaining a larger population on Moon, Mars, wherever, I imagine this quickly becomes a less than good pastime going around doing that. So has anyone looked at pollinators for Mars?
Thank you. That's really, I actually obviously thought about that myself. And I just have to add one thing I thought about when you said that, because a thing we haven't talked about really is what you lift up in the beginning, like being enthusiastic about something and really enjoying food. And that is so important for us as humans, because food can mean so much for us and really make our day, can't it, when we get something tasty. And I know there are studies showing how important food is for people being away from home. So that is even another aspect of it. And so obviously handling plants can be an additional nice thing away from when you're away from home. So to my knowledge, there haven't been any studies or really much thinking about pollinators. We will likely need them because at least today, I think, is it 80% of our plants are pollinated? So insects have so many other services that we don't really think about, that we use. I think, if I don't misremember, there has been a study looking at insect flight in space and have shown that surprisingly, I don't remember which species it was, but they could sort of learn and how to correct for that environment, which is a bit amazing. And I assume that at least if you have some gravity, you could, there would be probably some plasticity within a species to learn how to correct. But then we have an additional thing on top of that we have not talked about, and that is the evolution of species in a totally different environment. And I expect that humans will do the same as all the animals and plants that we will take with us, because we will face another environment that we have to survive in. So there are different layers there, but it will certainly be important wherever we go and however it looks where we are going.
Jan Rutweger from the DLR. Obviously, pollinators may be important, but what we should also consider is the microgravity. The pollens don't sediment, so it will depend on. They will stay, in the air for much longer. So it will depend exactly on the plants that you look at. Some are really specific; they need specific insects in order to open the blossom. Whereas more by an open access, I think it may be even easier in space, in microgravity.
I just wanted to add on the multi-generational aspect. It's actually something that hasn't been investigated much in general for any species, whether it be insect or mammalian cells. And that is something that's very interesting because we have no idea what would happen after 10 generations in terms of the mutations. So,
yeah, thank you for the interesting talks as well. Is there anything also going on with crustaceans? Like you talk about insects, but there are some shrimps that you can transport very easily with the eggs, and then they can feed on cyanobacteria and so on. So in these MELISA concepts or things like that to increase the protein content, is there anything going on with these type of organisms as well on top of insects?
I can start with what I've seen. I don't think I've seen they have done some experiments, but not as for food, and looked at that. But we haven't really looked at that, so we focused on insects for different other purposes. And one of the reasons why also we didn't look into that so much is because we were thinking of terrestrial organisms that don't need water, so not that dependent on water anyway. But I know there are some studies, and obviously, who doesn't love eating crustaceans? So that could obviously be an interesting aspect as well. I don't know if you have a thought about.
I don't work with it myself, but there's a group of students, a cross-European group that is, I think they were supposed to launch an experiment with shrimp. I don't know if to the ISS or any other cargo ship, but yeah, I can look up the name on LinkedIn and let you know.
Yeah. Do you know if vegan regime was already investigated for long-term space too? Because potentially you avoid all the problem for food, animal or insect food production. So I don't know if there is a limitation that was already highlighted for astronauts in this kind of regime.
Sorry, I missed the first. Which regime?
Vegan regime.
Oh, okay. No, I don't know. And the reason we are interested in this is because, like we heard, there are animal proteins that sort of give us basically all we need as humans to grow. And the reason why we are interested in insects is not just that they give us food. So they could be a part of a system where they eat the stuff that we don't eat and convert that instead of getting shitloads of waste. They use the waste and they grow and we get really good proteins, fats, vitamins, and so on. So they have so much more than just food. That's why we think this could be something good. So, but I don't know if you have heard about vegan food.
I know that there's some vegan astronauts or vegetarian astronauts. I do think it should be looked into, but I agree that it will be more difficult if you have only one technology just to close the loop, as opposed to having insects or macroalgae plants. I think from the technological point of view, it's much easier just to grow plants, but then in terms of calorie, they're also way less dense when you compare with insects or meat. So then you get into a problem of volume and mass.
Thank you. These talks are inspiring. I have also a lot of ideas, particular to interactions, because you mentioned you are working also ecosystems, because you are focusing now on insects, but thinking about waste interactions, always this is depending on microorganisms. So this interchange also with others, I would say microorganisms and microbiology, always taken into account. Are there any studies, interactions also? Because I know the community of astrobiology also is more thinking on applied astrobiology, and this could be also linked because some of these organisms are very resistant against space conditions and could help.
Yeah. So I totally agree, and there are so much neat stuff that could be done. We are having a study at the moment that we have started an experimental study where we have 3 components. So that is one insect species, and we have one plant that we use to grow for human food, and where we're going to use the insect frass, the poo, as a fertilizer, and looking at how that changed the microbiota community in the roots of the plants and how it affects the nutrition and growth and so on of the plant. So we are trying to look at that, but we are just starting. So if there is anyone else with ideas, and I'm very happy to hear about that.
I have a question as well, because Joao, you mentioned that one thing that's really expensive is the medium to cultivate cells. Could you envision a system where you actually convert the waste into a medium that you can then use to cultivate cells?
Waste from?
From the astronaut, from the environment?
You probably could use it as a base, but you need to add a lot of other stuff, because you'll be lacking amino acids and potentially glucose as well. So we're working now with using brewer spent grain and brewer spent yeast as a base for our media. So you can use some of these waste streams, but you always have to supplement it with some additional compounds. Microalgae, for instance, they're a very good source of amino acids, and that's what the group in Japan has been working with. Yeah.
I have one for each, if that's okay. So for the first speaker, we talked yesterday, and thank you for mentioning the evolutionary concept. I wanted to ask, because you mentioned that there's a problem with the development of the insects, so should we focus on using species which are eatable already at the stage of larvae, and then have like separate population that will allow to, you know, grow further and lay eggs? Or is there any reason we should like each time let them mature for some reason? And for the second speaker, because you mentioned that the cells, when you take them, primordial cells from the animals, they will grow, and then they will plateau. So you need to find a way to make them like, to immortalize them. But I mean, I get some red flag in my head. Isn't that like a cancer cell? So how do you control so they don't get some weird mutations inside? Thanks.
Yeah, go ahead. Thank you. Yeah, and thanks for a nice talk yesterday. Yeah, that's a bit hard because we don't really know what's going to happen. But thinking about what we know at the moment, I would think it will be carried out approximately like it is done at the moment, where you have you focus on food production, and then you select individuals from that production that you use for mom and dads for the coming generations, and you secure that by having quite lots of animals going all the time in different life stages. And when you eat, yeah, how the selection is done. I think it's, so we do have selection experiments going for the moment where we do select on different parameters and see how effective that is. I assume you would select for individuals that do really well in the environment and produce lots of young that are healthy and good. So you would do a selection that would be slightly different than what you do here. And then when you eat them, I guess it depends on what kind of food you want, if they change the flavor or nutrition or so on. So that, I guess, stuff you sort out, hopefully mostly on Earth, and then hopefully we get a chance to try it out to see how it works.
Yeah, so thanks for the question. We get that a lot. So there's basically three sort of hallmarks to define a cancer or tumor cell. So it needs to be able to divide indefinitely, and that's what we have. But it also needs to be able to form tumors, and genomically it's quite unstable. So we can check for this, and of course I'm not doing that at the lab scale, but companies that are working at the large scale already, they do check for tumorigenesis of the cells and genomic stability of the cells. So that's part one. So no, it's not a cancer cell. Second, there was a study commissioned by the World Health Organization and FAO to look at the safety of these products. And they saw that, or they concluded that even if there's these type of mutations which are similar to cancer cells, they most likely do not pose a risk to consumers, because first, when you eat them, they'll be dead, cooked, and they'll be digested. And that's no different from the meat or the fish we eat nowadays. So there's no way we can say for sure that the piece of steak we eat, that there was no cell with a cancer mutation there. So, yeah.
For the use of insects in life support systems, I think the Chinese life support concept has always taken insects on board, and there has always been tests in their terrestrial studies, I think in Beijing University, also in the Lunar Palace tests, I think. Was there anything that you could learn from that, and what were maybe the barriers to not include it or include it in European concepts? Or in your topical team, did you discuss that, look into that?
Yeah, so we have seen those studies. I think it's mostly silk, what we call silkworms. They use them mainly for silk production, and then they use the pupae to cook for. I think it's part of the like a traditional food as well. So we have looked at and see what kind of data and results they have. So that it is incorporated in that. The reason why one of the reasons why we are not using that species is that it's not approved for consumption in Europe. So we want to work with insects that we are okay to eat now, but thanks.
Okay, I have a question to João also. Just back to the question with tumor cells and so on. Are you studying also stem cells? So this production, because for the immortality, there could be also some key elements. Are you doing some work on it?
Yeah, so the cells we use, so there's two major cell types we can use. So we would call them adult stem cells. So these are cells that are in the tissue. That's what we primarily use. So those would be called satellite cells if you want to produce muscle, or fibroadipogenic if you want to produce fat. So adult stem cells, those are the ones that have a growth limitation. But then you can use embryonic stem cells or induced pluripotent stem cells. There is a company in the Netherlands called Meatable. They use iPSCs, and they modify the cells to not to proliferate faster, but to differentiate faster. So what they achieved was, while our process would generally take 2 weeks to fully differentiate the cell, they can do it in about 4 days, either fat or muscle. So across the industry, you'll see companies using primary cells, cell lines, or pluripotent stem cells. Yeah.
Hi, really great talk, by the way, Zhao. So I was actually senior scientist at Val whilst we were developing all of that. The question I have for you, though, is about your bioprocess optimization and intensification. So you mentioned 500 liters of the bioreactor. As that circulates around, it's a fairly inefficient process, so you're going to have some filtration. Things you need to filter out are nitrogenous waste and probably lactate, but you've also got a lot of very valuable things like cytokines, growth factors, amino acids, which you need to keep in. Do you have an idea how you develop a suitable filtration system that can get rid of the stuff that you don't want, pellet it, for example, in a solid phase, and then recirculate the stuff that's just still useful?
We haven't thought that far, but yeah, that's something you would definitely need also to remove unwanted compounds like ammonia or lactate, indeed. I saw a presentation, I don't know if you were at the conference last week on cultivated meat in Maastricht, where they use an organic compound, zirconium phosphate, to absorb ammonia. So something like that could work for ammonia. But yeah, I mean, this is very much at the preliminary stage. Yeah.
Exciting, though.
Yeah.
I have another question about cultivating meat. So there is some evidence to suggest that when you culture stem cells in microgravity, they seem to proliferate more effectively. So there's some limited data on this, but do you think that it would be more efficient to grow steaks in space?
I found those papers initially, and I struggled to understand why. So one of the reasons could be that, you know, stem cells generally, or pluripotent stem cells generally, they'll be in the uterus, so in the placenta, within the placenta, in the amniotic fluid, so in sort of this free-floating state, which might be similar to what they find in space. I guess you'd need to test. So most likely the adult stem cells, the muscle progenitor cells, might have some troubles growing in space, while if you work with iPSCs, they might grow better, but we don't know. It would be great if they grow faster, though, or better. Yeah.
But what I found interesting about that was when you look at the human as a whole in microgravity, it seems that the stem cell proliferation is not speeding up. But if you are having the cells in isolation, it's sped up.
Yeah.
So it's a bit conflicting, but it might be because of this environmental impact that you are isolated from the rest of the body.
Yeah, I think probably growth would be better or could be better, but then I think you probably have problems with the differentiation. So whenever you want to make muscle out of it, fat, I don't know, but probably muscle would be quite tricky. Although when I was at the agency and we did some simulated microgravity experiments, we did not see a difference between 1G and simulated microgravity for muscle cells. I mean, but that's a two-time experiment, so I wouldn't really trust the data.
I thank you both for the very nice presentation. I think I already have the discussion with you, João, but I just want to bring it again. I don't know if you remember, but I was talking about if we are using cultivated meat, can we improve or even optimize this food system for the astronauts? Because you show we have that amount of protein, for example. What are your thoughts nowadays? Are we there, or are we super far?
If we can improve, I think it's, and I think the problem is that we first need to define what we need in space, whether we need meat or animal protein in general, and plants or vegetables and carbohydrates, and then I think we should start thinking about which technology to bring up. I do think cultivated meat can sort of, as I showed before with the plants, can break some boundaries. So instead of having only meat in one product, you can have sort of a superfood. And there we have plant scaffolds, but you can think about combining it with some insect powder or microalgae. So yeah, you have the potential to create different products that don't exist nowadays. So, and as we grow the cells, we can also feed them with molecules that we want. So we can supplement them with vitamin B12, D, C, A, you name it. So the cells will generally, well, some of these molecules, they will be taken up by the cells. So yeah, you can have sort of a fortified product.
We're not finished. Now it's my questions. I will not be too long, but one major thing we need to consider is for these long-term exploration missions, how do we convert the raw material, like you mentioned cooking, and okay, we might have the insects, but how do we take it from that state to actually food that you can eat? What kind of technology do you think is needed to do that process? Or is this something you would envision the crew to do themselves? What are your thoughts on this?
Yes, I should have the food technology person answering that question. I think if for people to be engaged and be part of their own cooking, I think is if it's in the right level, that's a good thing. I think to have something comforting and maybe cook not just for yourself but for others. So one part of that I think probably would be good. Then I envision that there would be technical help to both for insects when it comes to both heat up and sort of grinding and mixing, I guess, what you do with all the other food products that we need. So I can't see it's so much different compared to other food sources, really. As all food, you have the basic elements, you have to cook it and make it in the form with the spices and everything like other stuff that you want to eat. So I guess the difference might be that if we're talking about things that are alive, you have to, obviously, if you don't want to eat them alive, you have to kill them. So then it's the matter, how do you kill them? And today, what is regarded as the most sort of humane way of killing insects is that you cool them down and freeze them. because it mimics their their natural way of going to sleep in cold areas and die. So if there's a way of utilizing the coldness somehow in the environment and doing that, but then I think that's the whole other part of the technical issues that have to be developed with people that know their stuff when it comes to that. Mm-hmm.
Yeah, I think for the cultivated meat is somehow similar. So to collect your biomass, you would of course need to centrifuge and separate that from your culture media, and or either centrifuging or some sort of filtration to then produce the product. Yeah, you need some, I guess, some sort of extrusion mechanisms, or you maybe blend it with some plant protein, other components to create some sort of texture. If you use scaffolds, maybe you can get away with the scaffolds. For cooking, this is something we've given some thought back in the days when I was at ESA. So if some companies are actually growing fish or tuna and salmon, so in theory that you could eat raw. And I checked with them last week. The products that they produce is for consumption, like sashimi and so on. For meat, they would need to cook. You can think about sous vide, which most likely would work. And because you cannot use barbecuing, you can think about creating the grilled pattern with lasers. So we could give a steak the texture we know. And I think at the moment in the ISS there's an oven, so you can also think about using the oven to cook. So it is something we need to think about, but I don't think would be an obstacle for us.
I have a question. I don't think the microphone's on it. For the meat, is it necessary to cook it? Like for a health point of view, like does it release more nutrients when it's cooked? Because it should be sterile, right? There's no risk of sickness from it.
No, from a safety point of view, wouldn't be an issue. Is that more from the taste perspective? Yeah, but in most cases you would be able to eat it raw. Yeah. Yeah, but that would be conventional meat. Yeah. Yeah.
So then another question I have is, I think we are quite early on in the research that needs to be done to make this something that is applied. What kind of tools do you need that will help you in this feat? So I think there's a lot of stuff that still needs to happen in the terrestrial lab in terms of validating and studying the fundamentals. But what kind of platforms or things do you need to help you? Apart from money.
Always more money. No, you're totally right. As I said, there are so many things that needs to be done, fundamental things. So for us, I can see it's access to the ground-based facilities, able to continue with the work we're doing in our sort of home places and run that, but also access to different facilities and working with the people at the different facilities. That's what I see, what we need to do. And also people from other fields, they know the parts that we are not working with at the moment that we need to work together with to get ahead.
Access to facilities and space. Yeah.
Are there any facilities that are lacking currently? Is there a dream facility that you are looking for that doesn't exist?
All the stuff I've seen so far, not the facility in itself, but I wish that the instruments are made for larger things than they are at the moment, because the stuff we've been looking at is for very small samples. So more sort of animal-sized versions of instruments would be really, really good. So you could test different life stages and more animals at once, not just one or part of an animal, or also wants to get things more realistic, I would say, and to be able to do more in a shorter time.
So one of the things you might need is access to space as well. So I think one of the things I've noticed was this multi-generational aspect. Is there a particular number of generations you'd be interested in? It's just for us to understand the kind of platforms that you require and how we can push for them.
Yeah, so we, in the home lab, we run about approximately 5 generations per year. So you can do lots of study and study development, selection, and so on from generation to generation. So I think we can do quite a lot in 1 year. But if I think we brought that up several times during the day of making this real. We have to see the frames, sort of, what are the frames we go in, what are our boundaries? Our boundaries have to be positioned as well as we can think of where we want to go. How long is it? How long are we going to be there for if we're going to be away? What's the time limits? Or what's the times we are talking about? And what's the pressure and what do we need? Because that, I think, as well, would be good to know and to hear what that is, because then we could frame our experiments and studies accordingly. That would be actually quite nice as well, because I think most of us that work with this, we are interested in working with applied questions and problems and be a part of helping to solve these things. So any inputs on that will be good as well.
Okay, we are working on that. So I got a message from my colleague who's our plant scientist in the team, because she has been working with an expert group of scientists on a plant culture facility for the post-ISS era. And it would have been, this is why it was a shame that Veronica unfortunately couldn't join. We also have obviously an interest in plant and using plants for food, but there is still a lot of lack of understanding on the fundamental sides of how they grow in microgravity and in the space environment in general. So this plant facility definition team has worked on some requirements for a facility in space in the future, and they have concluded this now. And the main outcomes, or the main take-home message, is that what we need is larger growth volumes for plants, novel, totally autonomous analytical capabilities, and a new centrifuge for experiments in different gravity regimes, specifically for early growth stages of plants for future exploration. So this is part of the work that we will also propose for our next funding cycle, and hopefully we'll be able to develop. So if anyone wants to comment on those requirements, please go ahead. My colleague is also listening online, so she will be able to answer questions via the phone.
Well, I have not a question, but also a comment on this, because it is really highly needed to have bigger volumes, and I agree with that. But then I could tell you the best option is to go out on the moon, to try these facilities built up on the moon, using the in situ resources to build something up and then starting. Because if you're just sending a lot of masses in orbit and so on, you have an orbit, you have a moon, you have material there. Just go with the material science together, build up such kind of big facilities, and then start with the plants there. This is my suggestion.
Thank you. You probably have heard of the biomoon concept. I don't know if you were part of this. Yes, yeah. So actually, we have broken down the requirements of the biomoon concept into more feasible packages, let's say, and that's what we will propose for the next program period, to start working on that. Yes. So she's saying it includes plants, microbes, and fish, and a new paper was published this month on it. says more, let me know. Yeah, so I think this was really useful from the agency perspective, because there's a lot of things that I learned and I think we can bring back to our priority setting. There's definitely more things we can do to support this research stream. And I think we should work a little bit closer together as a community and also bring in the various expertises and disciplines, actually. It's a very cross-disciplinary field to try and work this together. So, yeah, I don't know if you had any concluding remarks or...
Yeah, maybe just, because this is something I noticed when I started working in space, which is not that long ago, but there's not so much research done on now. So we know that we lose muscle, bone density, but we don't know what happens to our gut system. So we don't know if nutrients are absorbed in the same way in microgravity. So I think there's some evidence that there's changes in the gut microbiome, but I don't think there's any study that looked at nutrient absorption in space. And I think from using tissue models or organoids, this would be fairly easy to investigate. And I think we should start there and then define what we want to eat in space or what's needed in space.
Yeah, I agree.
I can add one thing I thought about as well, we haven't mentioned, and that is the ability of creating other substances than food from the organisms that we grow. And I know there are studies being carried out to select different compounds and tailor both compounds and nutrient contents of different I'm just talking about the insects now, of course, but I'm sure there are for other organisms as well. And that makes, I think the value of this is much more than we understand at the moment, because we don't know so much. So I think there in the future, and we should keep our eyes open for and be a bit creative of things that we can create in the future. And I'm not talking about creating in some weird horror way, but there is sort of things I think we can develop that's already there, but we don't know it. And that can be medicinal areas or different types of nutrients that we can, utilize in a different ways, and make our immune system better from ingesting them, or make us more happy, maybe, or whatever we need. So I think there is a big future ahead, and we need to be open and creative, I think, together.
Yes. I just wanted to mention the colleague that I was talking for was Nicole Kaplan, in case you want to have some more information. So she's the expert on this domain. Sorry? Yes, so if you want her email address, please ask me. I will give it to you, and you can discuss further with her. So I think this concludes the session. Thank you very much for the very interesting discussions. I hope we can keep the momentum going for this topic, and we look forward to working for you more in the future. So, thank you very much.
Really interesting talks. Take care of this. Thank you. Thank you. She actually sent her email.
I cannot
Nicole
project
Copley
it.
yeah, but I think it's better not because of the YouTube.